Hello, everyone, and welcome to today's Tyson seminar series. Today, we have Dr. Camila Pizano joining us from La Universidad ICESI in Cali, Colombia, where she is an associate professor in the Department of Biological Sciences. She is an ecologist studying plant and soil ecology, including research in plant soil feedbacks, plant microbe interactions, seedling germination, plant functional traits, and fire ecology, especially in tropical dry forests. She received her bachelor's from La Universidad de los Andes in Bogotá, Colombia, and then went on to get a PhD in tropical ecology from the University of Florida with Kaoru Kitajima. She is also a talented biological illustrator with illustrations featured in a number of books and publications. Her scientific research is published in many ecological journals, including Ecology Letters, Ecology, and New Phytologist. Today, we're going to hear her tell us about Jansen-Connell effects in tropical dry forests, unraveling the interactive effects of drought and soil microbial communities on seedling survival and growth.
Many thanks, Filipa, for the introduction, and thanks for the invitation. I feel really honored to be here talking about my research, and thanks for that great introduction and summary. You saved me like 3 slides. I just wanted to mention before I start my talk that, as Filipa said, I've been working on plant-soil interactions in different ecosystems, most of them tropical, but also I did my postdoc in Alaska, in the tundra, amazingly enough. And just mention that I am the director of the herbarium at Universidad ICESI, and this has forced me to move into another type of research, particularly plant diversity. We just described a new species of plant in the Chocó biogeographical region in Colombia, and also a project on the plant endemic endangered plants of the high mountains of Colombia. So just mentioning a little bit about my research. As Filipa said, for the past 10 years, I've been working mostly on tropical dry forests in Colombia. I'm based in Colombia. We published this book in 2014, and I'm going to talk about a little bit about a national network we have of research on tropical dry forests. I just wanted to share with you my website in case anyone is interested in looking at my work. I really like the fact that I'm not only a scientist, but a scientific illustrator, and I teach illustration, and I love this combination of science and art. So if someone wants to check out some of my work, please do. That would be amazing. Okay. So today I'm going to talk to you about some research, very new research that I haven't published, very fresh results that I recently got from experiments that I have been setting up at Universidad ICESI. And I want to start with this very wide question that I think most of you have thought about, or at least listened to in one or more than one classes, right? And this is one of the main questions in ecology, and it's, how do so many species of trees coexist in tropical forests? We know that in the tropics, we have this really high diversity, biodiversity, but we still don't understand very well how can we have so many plant species coexisting at the local level. And when I say so many species, we have some tropical forests where we have 200 or even 300 species of trees. If you don't know how much a hectare is, it's more or less 2 football fields, right? If you look at this picture, what you notice is you don't see any dominating species. For example, when you go to temperate forests, sometimes you see pine species that are dominating, or oak species that are dominating, but you never see that in a tropical forest. What you have is a very high diversity of plants, and each species in very low abundance. So this has puzzled ecologists for a long time, as you can imagine, or as you know. This has resulted in many different research studies, different hypotheses. And today I want to focus on hypotheses that propose that there is not only a latitudinal gradient in diversity, but also a latitudinal gradient on the importance of biotic interactions. Biotic interactions and interactions between organisms. And some researchers proposed that these interactions between organisms are more important in tropical forests. And this is one of the reasons why there is such a high diversity of plants in the tropics. So which are these biotic interactions? We can think of interactions such as pollination, herbivory, seed dispersal, and also pathogens, right? And it's important to note we can have positive interaction, mutualistic interactions, such as pollination and seed dispersal, but we can have parasitic or not beneficial interactions, such as herbivory or plant pathogen, or even, some seed dispersers that don't disperse seeds, but actually predate on seeds, right? So we can have positive and negative interactions. There have been hundreds, if not thousands of studies looking at this interaction in different tropical forests. And some of the most famous studies in this framework are people who look at the Jansen-Connell hypothesis. As you might very well know, Jansen and Connell were two tropical ecologists who came basically to a similar conclusion, right? And which is this conclusion? That each tropical plant species has its own enemies, right? Or predators. In this case, we can think about herbivores, right? Herbivores. And what they noticed was if you have an adult tree, you're going to have a high abundance and a high number of these plant enemies, right? When this tree reproduces, it's going to produce a large amount of seeds. Most of those seeds are going to fall just beneath or underneath the parent tree, and some of these seeds are going to be dispersed. So because you have the parent tree here and a high abundance of seeds of this species, you're also going to have a high abundance of enemies, right? So what you're going to have is near the parent tree, you're going to have a high number of seeds, but a low survival probability. And as you get away from the parent tree, you're going to get a lower number of seeds, but a higher survival probability. So this framework or this hypothesis has inspired many studies, right? And you can search for many studies looking at how survival and growth of seedlings is compared, at different distances from the tree. However, if you only look at how species survive at different distance from the parent tree, you're not necessarily testing for coexistence. I'm going to explain why this is, just looking at distance and comparing how seedlings survive from the parent tree is not enough to test for coexistence. So first of all, which are the agents or the organisms that drive the Jansen-Connell effects? We can have herbivores, as I just mentioned. We can also have seed predators like peccaries, and we can have plant pathogens, right? The important thing is that for these processes to result in a high diversity, which I'm going to explain right now, these agents have to be species specific, but they are not all species specific. And why is this important? Because you can have a generalist enemy. For example, peccaries. Peccaries feed on many different plant species, right? And if they feed on different plant species, what you're going to have is a general effect. So in this diagram, you can see, this green tree, this green species attracts a certain enemy, which is generalist. And what you get is that all plant species are going to do equally poor close to this green species and equally well far away from the green species. So in this scenario, you're not going to have coexistence because all species do equally poorly close to the parent tree and equally well far away from the parent tree. So really, if you want to test for how these processes drive coexistence and maintain diversity, what you really need to have is each species having a species-specific enemy. So in this scenario, in this cartoon, you see the green species doing poorly close to its parent tree, but doing fine underneath the green tree, the red tree, excuse me. and the red species is doing poorly under the red parent tree, but is doing fine under the green parent tree. So what does this mean? So this means that if you want to have coexistence, this negative density dependence need to have two situations. The first situation is that enemies are species specific. So each plant species has its own enemies that attack more negatively this species than other species. And also each species has lower performance, either survival or growth or both, under the same species adult tree, as I tried to show in this cartoon. Okay, so as I said, most people, I mean, this framework has inspired many research publications and hypotheses, and many people have looked at the role of herbivores, above ground pathogens, and herbivores, seed predators in driving these patterns. But 10 years ago, people started thinking about below ground diversity, right? Because some scientists propose if we have just 1 gram of soil, we can have 10 to 50,000 species. And I'm saying species because maybe, you know, at the microscopic level, we don't have the same species concept, but we still have a lot of diversity. And in this huge diversity that you can see in this illustration, we have mutualistic organisms such as mycorrhizal fungi, such as nitrogen-fixing bacteria, but we also have a huge array of pathogenic organisms such as bacteria, fungi, or virus. And we also have neutral organisms that don't have an apparent effect on plants. As you can see in this illustration, we have a whole array of organisms that interact below ground, and many of these organisms can drive this negative density dependence or Janssen-Connell effects that I just explained. Okay, so how does this work? Each plant species associates with a different set of soil microbial species. So here you have species A on the left and species B on the left. And if you look at the colors and shapes of the soil microbes associated with the roots, they are different. And this can drive important ecological dynamics. Which organisms we can have? I just mentioned some, right? We can have beneficial organisms such as mycorrhizal fungi, which can be ectomycorrhizal or endomycorrhizal. We have nitrogen-fixing bacteria, we have worms, we have nutrient-decomposing bacteria, etc. And in the detrimental part, we can have fungal pathogens, bacterial pathogens, and lots of viruses. Something important to know is that, I mean, we might have these two very discrete groups, but really if we look at beneficial microorganisms, we can have them acting as parasites under some circumstances. So this is not so straightforward. We can have really a continuum between beneficial and detrimental organisms. The point is we have a huge diversity and we have positive, negative, and neutral interactions with soil microorganisms. Talking about some of these very important organisms, we can talk about arbuscular mycorrhizal fungi, or AMF. These fungi occur in 80 or 90% of plant species, particularly here in the tropics. And they are, as you see here in this slide, you see the fungi inside the root, and they produce spores that are dispersed in the soil, some of them by rodents. And we also have, for example, nitrogen-fixing bacteria that are mostly in legumes, and they produce these nodules. And both types of this positive interaction bring benefit to plants, such as nutrients, in case of nitrogen-fixing, nitrogen, in case of mycorrhizal phosphorus, and the plant gives sugar in return. On the other side, we have many soil pathogens, such as fungi and bacteria, and these might have a negative impact on different plant species. Okay. So this framework inspired what is called plant soil feedback dynamics by James Beaver. And what he proposed is that we have two species in this cartoon, right? Species A and species B. Species A might directly interact with species A and species B with species B. by intraspecific competition. Species can also compete directly against each other in interspecific competition. And importantly, each species is going to have a soil microbial community associated with its roots. And this soil microbial community is going to have a direct impact on each plant species, but it can also indirectly impact other plant species. So species A may impact species B by modifying the soil microbial community, and species B can also impact species A by modifying the soil microbial community. So we can have two different outcomes of feedback. First, we can talk about positive feedback. In this scenario, we have soil microbes coming from the red species or the green species, and in the y-axis, we have plant performance. In this first scenario, you see that each species does better with its own soil microbes. So red species does better with soil microbes from the red species, and the green species does better with soil microbes coming from the green species. In this case, we have positive feedback and we don't have coexistence because each species is going to favor individuals from its own species when it establishes. In the opposite case, we have negative feedback. Here we have again the same graph, right? But in this case, each species does better with soil microbes coming from the other plant species. For example, green species does better with soil microbes coming from the red species, and red species does better with soil microbes coming from the green. So in this case, we have negative feedback and we have coexistence because the establishment of one species is going to favor the establishment of other species. So how do we test for this feedback? What we do is we compare the growth of each plant species with its own soil microbes and growth with that same species with microbes from other plant species, and vice versa. As I explained before, we have to do this for each plant species. How would my results look like? If we have negative feedback, positive feedback, we would have positive bars, right? So in this case, we have positive bars and we wouldn't have coexistence. And if we have negative feedback, we would have negative bars and we would have coexistence. So if we summarize this, if we have positive feedback, species A is going to favor species A, and what we're going to have is a community that is monodominant, so dominated by one or few species, so we have low diversity. And if we have negative feedback, we're going to have species A favoring the establishment of other species in the neighborhood, and we're going to have coexistence and high species diversity. Okay. So this has been tested in several different ecosystems around the globe. This was one of the first papers published by Cleronomos in 2002. And very interestingly, you can see here that the abundant species all show positive feedback, whereas the rare species all show negative feedback in a Canadian oil field. So this is talking about herbaceous species in Canada. Going to the tropics, there are several important studies. I just want to mention one really cool study by Scott Mangan and collaborators, and he showed that feedback from soil organisms predicts plant species relative abundance in a tropical mature forest. What we have here in the top panel is the 6 plant species, right? And we see the strength of feedback. We see, first of all, it is all negative, right? But we see also that when they correlated the strength of feedback with the abundance of these plant species in the forest, they see this really neat correlation where we have, for example, species that have very negative feedback, such as such as BA, having a low abundance in the forest, and species that have less negative feedback, such as AA, having a higher abundance in the forest. What is really cool about this study is that they tested the same thing in the greenhouse and in the field, and they found that in the field, the organisms that were driving these feedbacks were mostly underground organisms, soil organisms. So this was also a seminal paper showing that these plant soil feedbacks really can explain the abundance of plant species in a mature tropical forest. Okay, so what do we have in tropical forests, right? To summarize this first part, we have many plant species per area. We have no plant species in high abundance. So all species, most species have very low abundance. Each plant species has its own species-specific enemies, and these enemies keep each plant species in low abundance. So we have that most species in tropical forests are rare. Okay, there was this really cool paper also published in 2017 by Lamana and some professors of WashU and other collaborators. In this really cool paper, you can see the world map, and they looked at how negative dependency changes across these forest plots of 25 hectares and above across the globe, right? So I want you to focus on two things. So first, there are some plots that are in the tropics, right? Here, yellow, orange, and green. And I highlighted this in this list of sites where they worked. And you can also see that in these graphs, you have the absolute latitude on the x-axis, and you have the strength of conspecific negative density dependence on the y-axis. What we see is that all the tropical plots are on the left of both graphs, right? So this means that tropical plots have very negative density dependence, and they also have very high plant diversity, which is what you would expect, right? So they show that at the global scale, really this density dependence can explain plant diversity. But I want you to look at something, or I want to highlight something, which is that most of these plots that were included in this study that are tropical forests, most of them are moist tropical forests. So these are sites where it rains 2,000 millimeters a year or above. So this is important, right? This is important because there are tropical ecosystems where it doesn't rain so much, and you might find a different pattern, which is what I want to talk today. Okay, so if we look at precipitation, really, we see that in this graph, you see monthly precipitation across the globe, and you see that in the tropics, you have some really wet sites, right? But you also have some drier sites, mostly in Australia and in Africa, but also in Northern South America. So really, the first question that I want to talk about is, you know, if plant soil feedbacks are also negative in these tropical ecosystems with a limited precipitation. If we see the same patterns of density dependence that have been shown for moist tropical forests. And I want to focus on tropical dry forests in Colombia. Well, first talk about a little bit tropical dry forests in the Americas. So America has more than half of the tropical dry forests of the world. These forests have at least 3 months of drought. This means a month of drought is a month of less than 100 millimeters. Some of these forests have 10 or 11 months of drought, but you have at least 3. This forest has a fragmented distribution due both to anthropogenic pressure, but also historic factors. And this really cool paper published in Science in 2016 showed that if you look at the composition of these forests across the Americas, you see really 18 floristic nuclei, which mean these forests, although they have lower diversity than tropical moist forests, they have high beta diversity. So you see a high change in the species that are dominant in these tropical forests in different parts of the continent. If we look at Colombia, so first I want to say in Colombia, mention the fact that we have this research network for studying tropical dry forests. And in this network, we have more than 20 institutions collaborating. We set up 16 1-hectare permanent plots. Here you have a map of Colombia. You see in this dark green the distribution of dry forest, and each one of these circles is one of these plots that we set up. Each one of these plots is 1 hectare, and in that hectare we measure and label every tree, palm, and liana that has 2.5 dBH and above. So I want to show you some really cool results, some some really cool differences between moist tropical forest and dry tropical forest. If you look, okay, so here in each circle, you're going to see the total number of species and the total number of individuals. The first thing to notice is that if you put together all the plant species of the 16 plots across the country, we have 623 species of trees, lianas, palms, and cacti, which is, you know, not a very large number. Interestingly, we have plots that go from 12 species. So we have one plot here that only has 12 plant species to a plot here that has almost 120 species per hectare. But if you compare that to tropical wet forest, you have half or less than half of the species that you find in tropical moist forest. Something other, a different aspect that is very interesting about tropical dry forest is that we have some sites like here in the Magdalena Valley, which is where I am right now exactly. And here in these plots, we have these two plant species, Trichilia carinata and Oxandra spinanta, which are very abundant. They are dominant in this plot. And interestingly, they are endemic, not just to Colombia, but to the Magdalena River. So we have this really interesting pattern where we have some plots where we will have actually some tree species that are dominating. And this is very different from tropical moist forest. So to summarize, we have a lower diversity in tropical dry forest compared to moist forest. And we also have some plots where we actually see plants that are very, very abundant, which is not happening in tropical moist forest. Or at least, well, we have some monodominant tropical moist forest that I'm not going to talk about today, but it's not the general trend in tropical moist forest. The other thing that is interesting is that if we look at the diversity of soil fungi, and this was a paper published in 2014, you see here on the right the sites where these researchers took their data from, and you see here and in the panel down here, you see the diversity of soil fungi across the globe. So the blue and green indicate low diversity, and yellow and red indicate high diversity. And you see that the pattern is similar to other organism diversity, right? Where you have diversity peaking in the tropics, but particularly in moist tropics. And this is what I want to highlight. If we look at soil fungi in tropical moist forests, we see very high diversity of mycorrhizal fungi, saprotrophs, and plant pathogens. And if we compare that to tropical dry forests, we see that diversity in tropical dry forests is 4 times or 3 times lower than what we see in tropical moist forests. So in summary, we have in tropical dry forests, we have lower diversity of plants, but also lower diversity of soil fungi and probably other soil organisms that were not studied in this paper. So my first question that I want to address today is, I really wanted to know if plant soil feedbacks between tropical dry forest plant species were also negative. And this is coming from, as I said in the introduction, I studied these feedbacks in Panama, in tropical moist forests, but also in montane forests in Colombia. And I found negative feedback in both of these forests. So I wanted to study now if these were the same type of dynamics in dry forests where we have lower plant diversity and we have lower fungal diversity. So to answer these questions, I want to first talk a little bit about which plants we find in tropical dry forests. So we have plants that associate with arbuscular mycorrhizal fungi. In this first group, we have 2 groups of plants. We have non-legume plants and we have non-fixing legume plants. And just to remember, arbuscular mycorrhizal fungi are endophytes that are inside most of the root and are benefiting plants for nutrients, particularly phosphorus. And some people have tested also beneficial water. So they also capture water from plants, and plants allocate up to 30% of the carbohydrates to these fungi. On the other hand, we have plants that associate with mycorrhizal fungi, but also nitrogen fixing bacteria. And here it's important to mention that these legumes, most of them are legumes, are very important plants in tropical dry forest. Actually, if you look at the most important tree family in terms of abundance in tropical dry forest across the Americas, legumes are the plants, the more important family, which is similar to tropical moist forest, right? I mean, we have legumes are always dominating in tropical forest. So this group of plants associate with nitrogen fixing bacteria, but they also have mycorrhizal fungi. Okay, so to try to get at feedbacks, I wanted to set up an experiment, and these feedback experiments always should have two phases. A first phase, an initial phase of soil training, and it's called soil training because in this first phase, each plant species is going to select its soil microbial community. So in this experiment, we had a common sterile soil. Sterile means without any soil microbes, right? To this sterile soil, we added soil collected from a dry forest. So we visited a dry forest close to ICESI, collected soil from different parts of this forest, combine it, and then use it in this experiment so that plant species, which were these 4 plant species, each plant species would select its microbial community. The 4 plant species were Ura crepitans, which is a non-legume, Hymenea curvaril, which is a non-fixing legume, and we had the 2 nitrogen-fixing legumes, Piteselebium dulce and Pseudosamanea guachapela, for a total of 4 plant species. In the second phase of the experiment, we had these 4 plant species that I just mentioned, right? Non-legume, non-fixing legume, and nitrogen-fixing legumes. And then each species grew with its own soil microbes or soil microorganisms coming from each of the other 3 species. So for example, if we had Ura crepitans grew with soil microbes coming from its own roots from the first phase, or soil microbes coming from Hymenea, from Piteselebium, and from Pseudosamanea. So basically we had the 4 plant species. Each 4 plant species was growing with its own soil microbes or soil microbes coming from each of the other 3 plant species. And just to recall, how do you measure feedback? You measure it comparing the growth of each plant species with its own soil microbes and growth of each species with microbes from the other, in this case, the other 3 plant species. And just so that you remember, we could have negative feedback, so that would be bars going below 0, and positive feedback, bars going above 0. Okay, so remember that I just explained that I mentioned several studies in the tropics showing that feedbacks in the tropics and tropical forests are mostly negative. However, in this experiment, so what we have here is the feedback value in the y-axis, and in the x-axis, we have all possible combinations of the 4 plant species that we studied. So Ura and Hymenea, or every combination. So you see that some bars are going up, some bars are going down, and some bars are not going anywhere. But importantly, none of them were significant. So basically what this means is that we found no evidence of negative of plant soil feedbacks in this dry forest. And this is really surprising. I mean, I was expecting to find negative feedback, just as I have studied in other tropical forests, and as the literature reports for other tropical forests. So why is that we don't see a plant soil negative feedback? Maybe there's, we have mutualistic, which were the soil microbes that I presented, nitrogen fixing, we have a mycorrhizae, but we also have pathogenic. So maybe they're just, they're just evening out. So there's a neutral effect when you have mutualists and pathogens together, which doesn't make a lot of sense because in other forests, tropical forests, you have mutualists and pathogens too, but that might be a possibility. Another possibility is that, as I told you, studies have reported we have a lower diversity of organisms, right? And this might be in part because of these drought conditions, of these dry seasons that result in a lower diversity of soil organisms. Another possibility is we lost diversity. These forests, as I just mentioned, are all fragmented, many are degraded. So maybe we had a very high diversity of soil organisms and we have lost that diversity. And so this has resulted in no soil organisms that have a specific species-specific effect. And finally, it's possible that in dry forests, biotic interactions are not as important as abiotic pressures, and in this case, drought. And I want to focus in this aspect. I just mentioned that tropical forests are very fragmented. Actually, tropical forests are among the most endangered biomes in the neotropics. And there's a big question of what is going to happen to these forests with changes in precipitation. And we are already seeing changes in precipitation due to climate change in these forests. So how can precipitation change? There are different options, right? So in this graph you're seeing here, we have in the A panel the normal seasonal rainfall in a dry forest where you have dry seasons and wet seasons, right? So one first possibility is that you have reduced rainfall. So you have the same dry and the same rain seasons, but there are some rain seasons where it doesn't rain what it used to rain before. This is one possibility. Another possibility is that you have this is the first. The second is that you have changes in the dry season length, or in the rain season length. So for example, here in the sea, right, you have these three normal dry and wet seasons, but then this wet season is shorter, right? So you have changes in the length of the dry season or the rainy season. You can also have a change in timing. So you can have the same amount of rainy and dry seasons, but they change during the year. Or you can have also reduced precipitation. So you have the same rain season, but it rains less in several rain seasons. Or you can have a multi-year drought, which is, for example, what we experienced from 2014 to 2016, which we have one of the driest El Nino events for the past 20 years. Today, I want to focus on these two options: reduced precipitation and changes in the frequency of the rain. And I want to focus on these two possibilities. I mean, you could focus on any of them, but I chose these two possibilities to explore how would changes in precipitation impact plant-soil interactions in dry forest. If you see this picture, this is the dry forest in the rainy season. You see all the forest is green. Trees are producing new leaves. And in the dry season, you see many trees without leaves and just plants kind of surviving, but not doing very well. Okay, so first I want to show you some data we got from the field. Again, here we have the map of Colombia. The dry forest in this map is in yellow, and we have the 16 plots that I told you about. But for this research that I'm going to show you, for this data, we only selected 7 plots. These stars you see here, the 7 plots. And in each of these plots, we have the 1 hectare plot, which is divided in 100 subplots. From those 100 subplots, we only took 45, and we took 1 soil sample in each one of those 45 subplots. These soil samples, 15 centimeters in depth, were separated soils from roots, and the roots were processed in the lab to measure arbuscular mycorrhizal colonization. What does this show us? This shows us how active these fungi are in the soil, because it's an indication of how much they colonize the roots. Okay, what we find here in the x-axis, we have the evapotranspiration of the sites. So the higher the evapotranspiration is, the drier the site. And in the y-axis, we have the colonization of mycorrhiza in the roots. So you see here, it's only 7 points, right? The 7 plots that I just explained. But we see this negative relationship where in drier sites, we have a lower activity of mycorrhizal fungi. So then I wanted to explore at the species level. I mean, if we test at the species level, how do different soil microbes may, if they hinder or they help plant tropical dry forest plants facing drought, right? So I wanted to do a greenhouse experiment working with different plants and plants that associate with different soil microbes. So for this experiment, I had 2 soil treatments. I had a sterilized soil that had no soil microbes, and I had a soil that had microorganisms from a tropical dry forest. I had 3 different irrigation treatments. So the control really was the equivalent to what it rains in a dry forest, which is 1,600 millimeters per year. The reduced frequency was the same amount of water, but only every other week. And the reduced precipitation was half the water. So plants were watered the same 2 times a week, the same as the control, but half the amount of the water. And then I had 9 plant species of the same groups that I talked in the previous experiment. So I had 4 non-legumes, 2 non-fixing legumes, and 3 nitrogen-fixing legumes. So this is to set up the experiment. I had a common sterilized soil. To that sterilized soil, I added soil that I brought from a dry forest again. That soil was either sterilized, so had no soil microbes, or was live, so it had all soil microbes. I had the 3 irrigation treatments that I just explained: control, reduced frequency, and reduced precipitation. And I had the 9 plant species that I just explained. This is how the experiment looked in the greenhouse. Okay, so the first really cool result is that reduced precipitation had a stronger negative effect on plant growth. So we see in this graph, these are all plants together, and we see that the control grew significantly better, but they did worse with reduced precipitation than with reduced frequency. So this means that it's not the same, that changes is not just reduced precipitation. If we have different changes in the precipitation regime, we're going to have a different impact on plant species, which I think is an interesting first result. The second, okay, so how did non-legumes respond? So what you're going to see 3 slides that have the same graph. So what we're going to have here is on the x-axis, we're going to have the control, the reduced frequency, and the reduced precipitation. And we're going to have plants growing with a live soil, so with soil microbes, and in a sterile soil without soil microbes. So this first group of species, the non-legumes, really grew the same with or without soil microbes. I mean, of course, drought really reduced growth of this plant, of all plant species, but you don't see an effect of microbes for this group of plant species. However, if we look at the non-legume species, you see non-fixing legumes, sorry, we see this really interesting pattern where these species grew both in the control and with both drought treatments. They grew better without than with soil microbes. So you see this significant higher growth with sterilized soil than with live soil. So this might mean that you have an effect of pathogens, or it may also mean that you have a negative net effect of both pathogens and mutualistic organisms such as mycorrhizal fungi. This was the same trend for fixing legumes. So we have these 3 plant species. You see the same pattern as the slide before. So these plants again grew better without soil microbes, so in the sterile compared to the live soil, both in the control and in the drought treatment. What was really cool too is that for these nitrogen fixing legumes, we saw that, okay, here we have the 3 plant species, Pithecoloium, Pseudosamanea, and Samanea. And the first bar is the control, and then the 2 other bars are drought. And if we look at nodule dry weight, so how much nodules they form, the nodules are the ones that show us nitrogen fixing. So we see that drought really basically eliminated or significantly reduced the capacity of these plants to fix nitrogen. And we also find this interesting correlation between the nodule dry weight of each plant and the total dry weight of the plant. So basically nitrogen fixing legume growth dependent on nodule formation. So the plants that were able to form nodules grew better than the ones that didn't form nodules. So to summarize a little bit, legumes. So we know that the non-legumes really show no response to soil microbes, but legumes show these very interesting results to soil microbes and drought. So first, they had lower growth under drought. Second, nitrogen fixing really was minimized due to the effect of drought. Third, they were negatively impacted by soil microbes, both in the control and in the drought treatment. So that might be, the effect for probably of pathogens, or maybe some mutualistic organisms such as bacteria, nitrogen fixing bacteria, or mycorrhizal fungi might be pathogenic for these plant species. Okay, so to conclude, what I want to say is first, dry tropical plant species are more negatively impacted by changes in the amount than changes in the frequency of precipitation. This is interesting because the prediction of climate change is that different dry forests are going to get different, changes in precipitation in the future. Second, contrary to studies in other tropical forests, soil microbial communities don't seem to have a species-specific effect on tropical dry forest plants, and that's why we didn't detect any feedback dynamics. Soil microbes appear to have no effect on the growth of non-legume seedlings. Soil microbes hinder the growth of nitrogen fixing and non-nitrogen fixing legumes, regardless of water treatment, so in control and also under drought conditions. And finally, drought reduced nitrogen fixation, which hindered growth of nitrogen fixing legumes. So nitrogen fixing legumes were, growth was hindered both by reduced nitrogen fixation and by drought when there is water limitation. Okay, so future direction. There's a lot to do, as you can imagine. Every time you do research, you have more questions than answers, really. So first, I want to continue studying these plant soil feedbacks of different plant species, abundant, rare, legumes, not legumes in tropical dry forest, and different soil microbes such as mutualistic organisms, pathogens, ecto versus endomycorrhizae. I didn't mention it, but tropical dry forests are very interesting because we have many plants with ectomycorrhizae and also nitrogen fixing bacteria. Field surveys, we're doing that right now to explore soil microbial diversity across tropical dry forests that have different conservation status, but also precipitation regimes. And finally, effects of soil microbes on different plant species, abundant, rare, legumes, non-legumes, etc., across contrasting precipitation regimes. It is interesting because maybe some of these soil microbes, so, the results suggest that pathogens are important, but maybe some mutualists may also become parasitic under some precipitation regimes or under drought stress. With that, I want to thank all of you for listening to my talk, and many of my collaborators, Scott Mangan, who has been my collaborator for 20 years, Jennifer Powers, the network of research dry forests in Colombia. I mentioned it's more than 20 institutions. We are about 40 researchers directed by Roy Gonzalez, another of my great collaborators, and of course, many students in ICESI and Universidad ICESI, which has given me the facilities and part of the money to do my research. Thanks to everybody. And with that, I would be more than happy to answer any questions.
Thank you, Dr. Pizano, for that amazing talk. So just so you know, everyone, you can relay your questions on the YouTube chat, so I can relay them to Dr. Pizano. Our first question is from Susan Louis. Susan asked, did you draw the cover illustration, I guess, for the Dry Forest book? And in those lines, Dr. Pizano, how long did it take you to draw that illustration?
Good question. Yes, thank you. I did, well, this is going to take me too long to go back, but yes, let me just, hold on. Yes, thanks for the question. I did do the cover of the book of dry forest and also the book on the red list of endemic plants of the Paramo, which we just published 2 months ago. So yes, you can download these books. I can, if I share a website, Jan, will you be able to share the website with everybody?
Yes, I can do that.
Okay, so if you want, I can share the site where you can get the book, any of these 2 books, and this illustration took me about a month. If you see here, I have plant species, but also animal species. They're difficult to see. There are 6 animal species in the illustration, but they are all dry forest species. So it took me a month. I mean, not working all day, right? But a month on and off to do this illustration.
Okay, the next question is from the Adventure Breeze. So, is plant invasion more common in tropic dry forest versus moist forest? And the Adventure Breeze is curious if lower diversity of mycorrhizae in dry forest prevents establishment of non-native species.
Excellent question. We don't know. Okay, so the first question, I mean, I don't know. There might be studies that compare invasive species in moist tropical forest and dry forest. I don't know of any studies that compare, but they might exist. Both have high invasive, how do you say? They are both invaded by many species. Dry forests have a very particular condition, which is that they are fragmented, as I explained in my talk. But also most of them occur in relatively good soil. So many are surrounded by agriculture and pastures. So some of the most aggressive invasive species in dry forests are African grasses, but the same has been reported in some moist tropical forests. So I don't think it's unique to tropical dry forest, but that is a very good question. And the other question was if you have a low diversity of mycorrhizae, Jan?
Yeah, it was the Adventure Breeze is curious about if lower diversity in mycorrhizae in dry forests prevent the establishment of non-native species.
We don't know, and that's a very good question. And we don't know if there's a lower, I mean, this paper that I talked about, yeah, it appears that there is a lower diversity of mycorrhizae in dry forest, but we don't know. It might be the case. I mean, it might be the case that it allows or hinders the establishment of invasive species. Very good questions. Great ideas to do research.
The next question, Abby Gorline asks, as someone who wants to incorporate both science and are into the career somehow, so I have 2 questions. One, how do you make time to do the work on both, since they are both time consuming? And also, do you have any general advice for someone starting out in art and science?
Definitely. Okay, so the first question, when the scientific career, you work a lot and it's great. We love doing science. But art is always a great way of relaxing. So it's also work, but it's relaxing. So I do most of my scientific illustration during the weekends or during the night. And it's always, I mean, it has always been a great way of disconnecting, of relaxing from my other work. So I have never seen it as work. I see it as a relaxing activity, and I have always done it parallel to science. And I think it complements in a great way the science that we do. And an advice would be, as I always tell my students, just start and never stop illustrating, never stop. So what I do, I'm always illustrating something. I've been very lucky because since I started illustrating, I'm always illustrating something for scientists, for different people. So never stop doing it. You know, just as you exercise, just as you sleep, just as you eat, work on illustration, and also keep a very organized, you know, portfolio that you can show. I think that's part of you know, having a website, for example, or having Instagram is a way. It's a great way of showing your work, because if somebody contacts you, then you have your portfolio. You can show the different things that you have done. So that would be my recommendation. Do it, never stop doing it, and keep, you know, putting your work on this platform so that people can know your work.
Hey, next question, Philippa Tanford asks, with climate change, we expect some environments to get more and some less precipitation. As research have shown, this can change the outcome of plants' relationship with their associated microbes. Do you think this could affect the structure of the forest?
Definitely, definitely. So that, I mean, that was one of the reasons why I wanted to look at, or why I want to continue looking at feedbacks in dry forests, because as I mentioned, right, we see this pattern where you have drier sites, have lower diversity, and if you have into account that it's not only climate change, but it's also the fact that, you know, these ecosystems, all ecosystems are being fragmented and degraded. What we see is, you know, the greatest possibilities that we're gonna lose diversity in these soil organisms. As we lose diversity in these soil organisms, we're going to start losing these plant soil feedbacks that have shown to maintain diversity in different ecosystems. So yeah, definitely. I think, I mean, that's the big question that I'm trying to get at with this research, definitely. But for doing this, we need, you know, we don't have information on what is the base. So first of all, if we talk about tropical dry forest, in Colombia at least, and most of the Americas, they are very fragmented forests. So we don't have the baseline. We don't know what the diversity of the soil organisms or plants was before we degraded and fragmented this forest. So maybe we already lost, you know, part of this diversity, and maybe we are not seeing these plant soil feedbacks because we have lost this diversity. So yeah, definitely.
Next questions. David Esteban Garcia has two questions. The first one, was the live soil used in the experiment taken near mature trees, or from the same species studied, or from the random places in the plots? And the second question is, do you think there are pathogens that affect the growing of end-fixing bacteria in leguminous roots?
Okay, so the first question, I didn't understand. The species selected for the question is if the species selected for the experiment are also present in the plots. Did I understand that?
Let me read it again. Was the leaf soil used in the experiments taken near mature trees of the same species type, or from random places in the plots?
Oh, great question. Yeah, no. So we took it from random places in the dry forest, plot. And that's why we had the phase one of the training soil. So we wanted each plant species to select its own community of soil microbes. So that's why, yeah, we didn't collect from the parent tree species, but just from random places. And that's why we had the first phase of the training of the soil. And the second question he asked, if pathogens, Jan?
Do you think there are pathogens that affect the growing of end-fixing bacteria in the leguminous roots?
That's very probable. I didn't mention, but we have root samples from all plants from these experiments, and we are going to do DNA analysis to identify the main groups of microbes that are present in this plant species. So maybe we're going to see, and that's what I would expect, right? As I mentioned, maybe that negative net effect of soil microbes is due to pathogens. So that's what we expect to be able to see when we analyze DNA of these roots and identify maybe some pathogenic fungi that might, or bacteria, right, that are present in both non-fixing and fixing legumes, which might be driving this pattern of growing better without soil microbes than with soil microbes.
Okay, we don't have any more questions in the chat. So thank you again, Dr. Pizano, for your amazing talk. I hope we can see in the future more of your results. So thank you.
No, thank you. I hope I didn't talk too fast. I hope my English was understandable. I'm sorry, I haven't spoken English for a long time. So I hope I made myself clear. Thank you very much to everybody.